Dear students, after studying this module, you shall be able to know about the introduction of ion exchange chromatography, the principle of ion exchange chromatography, some of the exchange techniques, and the applications of ion exchange chromatography. For many years, it has been known that the clays include metal ions in their composition and that these ions can change places with the other metal ions in their vicinity. This observation was important to the understanding of soil chemistry. Zeolites, sodium aluminum silicates, have some properties similar to those of clays. Using this phenomena, natural and artificial zeolites became useful in removing calcium and magnesium ions from water. The calcium and magnesium ions dissolved in water are exchanged for the sodium ions from the zeolite, thereby providing a soft water. Ion exchangers have been created by combining a polymer and a functional group. Such ion exchangers have properties very similar to those of zeolites. Two types are available: cationic and anionic exchangers. Both of these use a resin as an insoluble inert support, but they use different functional groups in order to provide the type of exchanger necessary. The phenomena of ion exchange was first reported by two British agricultural chemists. In 1858, they proved that the soil can remove potassium or ammonium salts from water with the release of an equivalent amount of calcium salt. After the work of Gans in 1913, natural and synthetic inorganic cation exchangers were used for softening hard water. Modern ion exchangers resins were used first in 1935 by Adams and Holmes. These resins consist of three-dimensional networks of polymeric chains cross-linked with the short chains containing ionizable functional groups. The fundamental work on chromatographic separations by the use of ion exchange resins is mainly derived from the studies carried out in connection with the work of an atomic energy in USA. Definition: ion exchange may be defined as a reversible reaction in which the free mobile ions of the soil called ion exchange are exchanged for the different ions of the similar charge present in the solution. Most ion exchangers for practical use consist of an insoluble organic polymer into which a charged group has been introduced in some suitable manner. The backbone is generally a styrene divinyl benzene copolymer. Copolymers of the acrylic acid derivatives divinyl benzene are also frequently used. For use with the biological macromolecules, it is usual to introduce charged groups into the cellulose fibers. For several years, cross-linked dextran, Cefadex, has been used also as the carrier material. The most common properties of all ion exchangers are as follows. Firstly, they are almost insoluble in water and organic solvents such as benzene, carbon tetrachloride, ether, etc. They are complex in nature, that is, in fact, they are polymeric. They have active or counter ions that will exchange reversibly with other ions in the surrounding solution without any substantial change in the material. Principle: the principal feature underlying this form of chromatography is the attraction between the opposite charged particles. Many biological materials, for example amino acids and proteins, have ionizable groups, and in fact that they may carry a net positive or negative charge can be utilized in separating the mixtures of such compounds. The net charge exhibited by such compounds is dependent on their pKa and pH of the solutions in accordance with Henderson-Hasselbalch equation. Ion exchange separations are mainly carried out in columns packed with the ion exchanger. There are two types of ion exchangers: namely cation and anion exchangers. Cation exchangers possess negatively charged groups and these will attract the positive charged molecules. These exchangers are also called as acidic ion exchange materials since their negative charges result from the proteolysis of the acidic group. Anion exchangers have positively charged groups which will attract the negatively charged molecules. The term basic ion exchange materials are also used to describe these exchangers since positive charges generally result from the association of protons with the basic groups. The actual ion exchange mechanism is thought to be composed of 5 distinct steps. Firstly, diffusion of the ion to the exchanger surface. This occurs when quickly in homogeneous solutions. Diffusion of the ion through the matrix structure of the exchanger to the exchange site. This is dependent upon the degree of the cross linkage of the exchanger and the concentration of the solution. This process is thought to be the feature which controls the rate of whole of the ion exchange process. Exchange of ions at the exchange site. This is thought to occur instantaneously and is an equilibrium process. The more highly charged the molecule to be exchanged, the tighter it binds to the exchanger and the less readily it is displaced by the other ions. Diffusion of the exchanged ion through the exchanger to the surface. Selective desorption by the eluent and diffusion of the molecule into the external solution. The selective desorption of the bound molecule is achieved by the charges in the pH and or the ionic concentration or by the affinity elution, in which case an ion which has a greater affinity for the exchanger than has the bound molecule is introduced into the system. Cation exchangers. A cation exchanger is a high molecular weight, cross-linked polymer having sulfonic, carboxylic and phenolic etc. groups as an integral part of the resin and an equivalent amount of cations. Thus, a cation exchanger is nothing but a polymeric anion to which the active cations are attached. In these cation exchangers, the hydrogen ions are mobile and exchangeable with the other cations. The anions like carboxyl, sulfides, oxides remain attached to the resin network. When a cation exchanger is kept in a solution of a salt, some of the hydrogen ions of the resin enter the solution and an equivalent amount of the cations of the salt gel attached to the resin. Anion exchangers. An anion exchanger is a polymer having an amine or quaternary ammonium groups as the integral parts of the resin and an equivalent amount of the anions such as chloride, sulfide, sulfate, hydroxy, ions etc. These anions are mobile and exchangeable. Regeneration. Anion exchangers are generally supplied in the form of salts. Amines in particular are stable only in this form. On the other hand, cation exchangers can be converted into the H+ form by treatment with the aqueous acid followed by washing the ammonium base having the hydroxyl groups from the strongly basic anion exchanger on treatment with sodium hydroxide, while the weakly basic anion exchangers are converted into free amines. Whatever the form is in which the resin is to be used, it should always be submitted to the full cycle of charging and discharging before the use. Carrying out this step enhances its effective capacity. Ion exchange column used in the chromatographic separations. The principle of ion exchange chromatography is based upon the simple fact that the different cations or anions have different capacity to undergo exchange reaction on the surface of a given exchanger. The capacity of an ion to undergo exchange reaction has been found to depend upon the charge and the size of the hydrated ion in the solution. Under similar conditions, the capacity has been found to increase in the charge on the ion, that is the valency on the ion, but has been found to decrease with the increase in size of the hydrated ion. If we compare other hydrated ions of the same size, it is observed that the ionic charge plays an important role to determine their capacity to undergo exchange reaction. According to this view, the capacity amongst cations has been found to decrease in the order. Thorium 4 plus is greater than aluminium 3 plus, followed by calcium and sodium ions. Similarly, the capacity among the anions of the same size has been found to decrease in the order of phosphate, sulfate and the iodide ions. If we compare the ions carrying the same charge, the size of the hydrated ions play an important role to determine their capacity to undergo exchange reactions. Thus, amongst the univalent cations, the capacity has been found to decrease in the given order. Amongst the double charged cations, the capacity has been found to decrease in the order of from barium to magnesium. Amongst the univalent anions, the capacity has been found to decrease in the order where the iodide is maximum, followed by nitrate, bromide, cyano, chloride, hydroxy and the fluoride ions. If the active polyvalent ion in the resin is to be exchanged with the ion of a lower valency, the exchange has been found to be favorable by using much higher concentration of the solution. The quality of an ion exchange resin is determined by its capacity, which in turn depends upon the total number of the ion active groups per unit weight of the material. Greater the number of ions, greater is the capacity of the resin for the exchange process. The efficiency of the resin has been found to depend upon the degree of cross-linking. That is, greater the cross-linking, higher is the efficiency of the resin. Selection of suitable systems. It is useful to classify ion exchangers according to the pK value of their ion groups. However, the given table shows the simplified survey of the working ranges. For example, for a strong acid, the ionic groups are sulfide and they are suitable for the acidic and the alkaline chromatographic medium. Furthermore, the others have been described. For practical use, the dissolved material must be present also in the dissociated form if it is to be retained by the resin. It is most convenient to work at a pH where one of the components will be retained, while the other does not interact with the ion exchanger. The concentration of the participating ion of the opposite charge also plays an important role. The retained ions can be eluted with the aid of a high concentration of cations or anions without changing the pH and hence the dissociation relationships. Ion exchange capacity. An ion exchange capacity of a resin may be characterized by the number of ion active groups. This is called as the total ion exchange capacity. It is expressed in the terms of milli-equivalent per gram of ion exchange resin. The exchange capacity of a cation exchange resin is usually found in the laboratory by determining the number of milligram equivalents of sodium ions which are absorbed by 1 gram of a dry resin in the hydrogen form. A solution of sodium fluoride of a known concentration is passed through a column containing a known weight of the resin, and the acid eluting from the column is collected and titrated with the standard solution of an alkali. In V ml of an alkali solution of the strength N in milli-equivalents per liter, it is required to titrate all the acid eluted from the resin weighing W grams. Then capacity would be defined as V multiplied by normality divided by W. Similarly, the exchange capacity of an ion exchanger is determined by passing a solution of sodium nitrate of known concentration through the column. The concentration ions of the hydroxide ions in the solution eluting from the column is found by titration with standard solution of silver nitrate or acid respectively. Ion exchange techniques. Two techniques are generally used to ring solutions in contact with the ion exchange resins. They are, firstly, the batch method. This involves the single step equilibrium. The resin and the solutions are mixed in a vessel until the equilibrium is attained. The solution is then filtered off. The extent to which ions from the solution are exchanged for those on the resins depends upon the selectivity coefficient. In single step equilibrium of this type, only a small portion of the exchange capacity of the resin is utilized. The batch method is used for softening of the hard water and the production of the deionized or demineralized water. Column method. Essentially, ion exchange chromatography is a technique in which the separation of the components of the mixture is brought about by taking advantage of the different selectivity coefficients for the resin. The differences in the selectivity coefficient leads to the different migration rates on an ion exchange column. As explained in the chromatography, there are three important methods, that is frontal analysis and elution analysis and displacement development. The principles of these techniques are also applicable to the ion exchange chromatography. The apparatus used in the column method consists of a glass column fitted with the glass wool plugged or a sintered glass disc at the lower end. An ordinary burette could also be used. The resin to be used should have a small particle size. This provides a larger surface area for contact between the solution and the resin. In general, the diameter of the resin beads should be less than 1/10 of that of a column. Dry resin should never be used to pack the column. Slurry of the resin is made in distilled water and any fine particles are removed by decantation. The slurry is then slowly poured into the column containing some water. Care is to be taken that no air bubbles are formed in the packed column. To ensure that no air bubbles remain in the column and that the resin is uniformly distributed, the column is backwashed with the distilled water. Backwashing is done by running the tap from the bottom of the column. This removes the air bubbles trapped in the column and also removes any fine particles of the resin that might have remained in the column. The flow of water is stopped and the resin is allowed to settle. The excess water is then drained off. However, the level of water must never be allowed to fall that of the surface of the resin, as otherwise the resin may dry up and the channels may be formed in the resin bed. In such cases, there will be incomplete contact between the solution and the resin when the column is subsequently used. Applications of ion exchange. Because of the particular selectivity forces of the material, separation with the ion exchange is used. Organic ions which form salts with the oppositely charged ions in another phase can be separated too. Almost all ion exchangers reactions require aqueous solvents. Some applications of the ion exchangers are as follows. Firstly, separation of the similar ions from one another. Ion exchange chromatography is being used to separate similar ions from one another because of the different ions undergo exchange reactions to different extents. Purification of organic compounds extracted in water. Many natural products extracted in water have been found to be contaminated with ions originally present in water. Those ions can be removed by using the ion exchange processes. Separation of sugars. This method was developed by Chaim and Zill in the year 1951. These sugars are first of all converted into borate complexes and then separated. Removal of interfering radicals. In the estimation of calcium or barium ions by the oxalate or sulfate method, phosphate ion is found to interfere. Therefore, its removal becomes necessary, which is achieved by passing a solution of calcium or barium ions having the phosphate ions through the sulfonic acid cation exchanger. Softening of hard water. We are aware of the fact that the hardness of water is due to the presence of calcium and magnesium ions and the other divalent ions. These ions may be removed by passing the hard water through the cation exchangers charged with the sodium ions. Complete demineralization of water. This requires the removal of cations as well as the anions. For the removal, the water is passed through an acidic cation exchanger when the metallic cations sodium, calcium, magnesium etc. are exchanged by the hydrogen ions. The water obtained from the cation exchanger is then passed through the basic anion exchanger when the anions commonly present in the water like chloride, nitrite, sulfate etc. are exchanged with the hydroxyl ions of the exchanger. The H plus and the OH ions which pass into the solution in exchange for the cations and the anions respectively combine to form the unionized water. Generally, sulfonic acid resin is employed for the cation exchangers while a strongly basic resin is employed as an anion exchanger. Separation of lanthanides. During the Second World War, the separation of fission products presented a major problem to the analytical chemist, especially the mixture of lanthanide, Sm, Eu, deuterium, cerium, Pr and the other transition metals as well as the mixtures of cesium and rubidium, strontium and barium and zirconium and niobium in the tracer amounts. To separate these, synthetic resins were employed in combination with the technique of chromatographic elution. When the solution having the mixture of lanthanides is passed through the column packed with the particles of suitable ion exchange resin, the cations present in the solution undergo exchange with the hydrogen or any other cation that may be present in the ion exchanger. The cation having the maximum capacity to undergo exchange reaction gets held near the top, while the other cations get held further down in the column in the order of their decreasing capacities to undergo the exchange reaction. When elution is done with the citrate buffer solution, the lanthanide citrate complex ion having the maximum preference for the eluent comes out first, while the La3 plus citrate ion complex having the minimum preference comes out last. Separation of actinides. The ion exchange chromatographic technique has been played a unique role in the discovery of trans-plutonium elements in actinide series. In the actinide series, the elution occurs in the reverse order of the atomic number due to the actinide contraction, and this proved to be the only way for identifying these elements. Some of them having been produced only in the atom quantities. It may be said that their isolation would have been impossible by any other separation method. Separation of amino acids. Ion exchange chromatography has been used to separate the complex mixture of 18 amino acids obtained by the acid hydrolysis of proteins, hence can be extended to the various investigations required in the field of forensic chemistry. Preparation of pure reagents. Carbonate is invariably present in the solutions of sodium hydroxide used for the volumetric determinations. They give rise to the errors in the acid-base titrations. The simplest way to remove the carbonate by allowing the solution to pass through the column of strongly basic anion exchange resins in the hydroxide form when the carbonate is absorbed, releasing an equivalent concentration of the hydroxide ions. Hydrometallurgy. Many metals are recovered and purified commercially by ion exchange. Examples include uranium, thorium, lanthanides, actinides, gold, silver and platinum. In some cases, the scale of operation is relatively small, example lanthanides, but the intrinsic value of these metals is very high. In addition, the separation and recovery of the trace amounts of the toxic metals from effluent and waste stream is an important application from the environmental considerations. To summarize, the phenomena of ion exchange was first reported by two British agricultural chemists in 1858. They provided that soil can remove potassium or ammonium salts from water with release of an equivalent amount of calcium salt. Ion exchange may be defined as a reversible reaction in which the free mobile ions of a soil called ion exchange are exchanged for different ions of similar charge present in the solution. The principal feature underlying this form of chromatography is the attraction between the oppositely charged particles. Ion exchange separations are mainly carried out in columns packed with an ion exchanger. There are two types of ion exchangers, namely cation and anion exchangers. The principle of ion exchange chromatography is based upon the simple fact that the different cations or anions having different capacity to undergo exchange reactions on the surface of given exchanger. Two techniques are generally used to bring solutions in contact with the ion exchange resins. They are batch method and the column method. Applications of ion exchange chromatography includes separation of similar ions from one another, purification of the organic compounds extracted with water, separation of sugars, removal of interfering radicals, softening of hard water, complete demineralization of water, and hydrometallurgy.